Your blood sugar doesn't spike by accident. It rises because a system failed to regulate it. Not one organ, not one hormone, a system. Your pancreas releases insulin. Your liver controls glucose output. Your muscles absorb sugar. Your gut decides how fast carbohydrates hit your bloodstream. And your stress hormones can override all of it. When that system slips, numbers climb. Fasting glucose rises. A 1C creeps up. Insulin stays elevated. Most people are told to manage it, but here's what rarely gets explained. There are plants, backed by clinical research, that influence these exact pathways. Plant number 4 on this list can literally block your ability to taste sweetness and reduce sugar absorption at the same time. And plant number 13 has been shown in trials to significantly blunt post-meal glucose spikes by slowing carbohydrate breakdown before sugar even enters your bloodstream. This isn't about replacing medication. It's about understanding leverage inside the system. If you care about how metabolism actually works, subscribe, because we're breaking this down properly. Let's start. 1. Bitter melon, the insulin-acting plant. Bitter melon is one of the most researched plants in the world for blood sugar regulation. It grows across Asia, Africa, and parts of the Caribbean. It looks like a wrinkled cucumber with a rough surface, and it tastes exactly like its name suggests. Intensely bitter. That bitterness reflects its chemistry. Bitter melon contains several bioactive compounds, including charantin, vicine, and polypeptide P. These compounds have been studied for their potential glucose-lowering effects. What makes bitter melon unique is that it appears to act in multiple ways simultaneously. First, some of its compounds may act in an insulin-like manner. Polypeptide P in particular has been studied for insulin-mimicking properties, meaning it may help move glucose from the bloodstream into cells. Second, bitter melon appears to reduce glucose production in the liver. The liver is responsible for releasing stored glucose into the bloodstream. In insulin resistance, this release can become excessive. Bitter melon may help moderate that output. Third, it may enhance glucose uptake in muscle tissue. Muscle is one of the largest glucose sinks in the body. Improving how muscle cells absorb glucose reduces circulating blood sugar. Several human studies have shown reductions in fasting blood glucose with bitter melon supplementation. Results vary depending on dose and preparation, but the pattern is consistent enough that it continues to attract research interest. Now here's the important part. Bitter melon is not pharmaceutical insulin. It is not a replacement. Its effects are milder and more gradual. But for individuals working on overall metabolic health, it may offer supportive benefits. Preparation methods matter. Traditional use includes stir-frying slices with strong spices, juicing the raw fruit, very bitter, drying and powdering, standardized extracts in capsules. The bitterness is strong enough that some people avoid it entirely. But bitterness in plants often correlates with bioactive compounds. In herbal traditions, bitterness has long been associated with metabolic stimulation. The larger lesson with bitter melon is this: plants don't usually act through a single pathway, they influence systems. And bitter melon appears to influence several glucose-regulating pathways at once. 2. Fenugreek, slowing sugar at the source. Fenugreek works differently. Instead of acting like insulin, fenugreek influences digestion. The seeds of fenugreek are rich in soluble fiber. When consumed, this fiber absorbs water and forms a gel-like substance in the digestive tract. This gel slows gastric emptying, meaning food leaves your stomach more slowly. It also slows carbohydrate breakdown and glucose absorption in the small intestine. The result? A more gradual rise in blood sugar after meals. That matters because repeated sharp glucose spikes contribute to insulin resistance over time. Fenugreek also contains an amino acid derivative called 4-hydroxyisoleucine. Some research suggests it may stimulate insulin secretion in response to elevated blood glucose. So fenugreek works in two complementary ways. It slows the entry of glucose into the bloodstream. It may enhance insulin response when needed. Several clinical studies have shown improved fasting blood glucose and better long-term glycemic markers in individuals consuming fenugreek regularly. What makes fenugreek especially interesting is how practical it is. It's inexpensive. It's widely available. It can be incorporated into meals. Preparation methods include soaking seeds overnight and consuming them in the morning, grinding seeds into powder and adding to food, taking standardized capsules. It has a slightly bitter, maple-like flavor. Some people enjoy it. Others tolerate it. The key with fenugreek is consistency. Its effects are not dramatic overnight. They accumulate over time. It also highlights something important. Not all blood sugar support requires stimulating insulin. Sometimes, the smarter strategy is simply slowing the rate at which glucose enters the system. 3. Ceylon cinnamon, improving insulin sensitivity. Cinnamon is one of the most widely studied spices in metabolic research, but we need to distinguish between types. Cassia cinnamon is common and inexpensive, but contains higher levels of coumarin. Ceylon cinnamon contains far lower levels and is generally preferred for daily use. Research suggests cinnamon may improve insulin sensitivity. That means your cells respond more effectively to insulin already present. When insulin sensitivity improves, the pancreas doesn't have to work as hard. Lower circulating insulin often corresponds with better metabolic health. Cinnamon appears to enhance insulin receptor signaling. In simplified terms, it may improve the communication between insulin and the cell membrane. Some studies show reductions in fasting blood glucose and modest improvements in A1C levels with consistent cinnamon supplementation. The effect size varies. Not every study shows dramatic changes, but the trend across multiple trials suggests real metabolic influence. Mechanistically, cinnamon may also slow carbohydrate digestion, reduce oxidative stress, improve lipid profiles. Cinnamon's strength lies in accessibility. It can be added to coffee, mixed into yogurt, sprinkled on oatmeal, taken as capsules. Typical research doses range from 1 to 6 grams per day. It's not a cure, it's not aggressive, but it is sustainable. And sustainability matters far more than intensity. 4. White mulberry leaf, blocking the breakdown. White mulberry leaf works differently than the previous 3. Its key compound, DNJ, deoxynigeromycin, inhibits alpha-glucosidase, an enzyme responsible for breaking down complex carbohydrates into simple sugars. When that enzyme is inhibited, carbohydrates are digested more slowly. This reduces the magnitude of post-meal glucose spikes. Think of it like a speed regulator. Instead of sugar flooding into the bloodstream rapidly, it enters gradually. Several human studies have demonstrated reduced post-meal glucose responses when mulberry leaf extract is taken before carbohydrate-rich meals. Unlike some herbs that require weeks of build-up, mulberry leaf's enzyme inhibition works relatively quickly. It is often consumed as tea made from dried leaves, standardized capsules, extract powders. Mulberry leaf does not stimulate insulin. It does not mimic insulin. It moderates carbohydrate processing. And for individuals who struggle specifically with post-meal spikes, that mechanism can be highly relevant. Notice something about these 4 plants. They don't all do the same thing. Bitter melon influences insulin signaling and liver glucose production. Fenugreek slows digestion and supports insulin response. Cinnamon improves insulin sensitivity. Mulberry leaf blocks carbohydrate breakdown. 4 different mechanisms, 4 different entry points into the same system. That's important, because blood sugar dysregulation is rarely caused by one problem alone. It's a network imbalance, and effective strategies, whether pharmaceutical, lifestyle-based or plant-based, often work best when they address multiple pathways simultaneously. 5. Milk thistle, protecting the liver's glucose control in glucose overnight. In insulin resistance, glucose overnight. In insulin resistance, the liver often releases too much glucose, even when blood sugar is already elevated. This is one reason fasting glucose rises. Milk thistle contains a compound complex called silymarin. Silymarin has been studied extensively for liver protection, but more recently, research has explored its role in metabolic health. Clinical trials suggest milk thistle may improve insulin sensitivity, reduce fasting blood glucose, lower inflammatory markers, improve lipid profiles. Why would liver support influence blood sugar? Because fatty liver and insulin resistance are tightly linked. When fat accumulates in liver cells, insulin signaling becomes impaired. The liver continues releasing glucose even when it shouldn't. Milk thistle appears to reduce oxidative stress in liver tissue, support cellular regeneration, improve insulin responsiveness. This doesn't act like insulin. It doesn't block sugar absorption. It improves the organ responsible for glucose regulation. That's upstream thinking. Milk thistle is typically taken as a standardized extract of silymarin. It has one of the strongest safety profiles among herbal supplements, though medical supervision is still recommended for individuals on medications. When blood sugar management includes liver support, outcomes often improve. 6. Berberine, the AMPK activator. Berberine is not a plant. It's a compound extracted from several plants, including barberry and golden seal, and it's one of the most researched natural compounds in metabolic science. What makes berberine unique is its effect on AMPK. AMPK is often described as a metabolic master switch. When AMPK is activated, cells increase glucose uptake, fat metabolism improves, insulin sensitivity rises, energy efficiency improves. Some pharmaceutical drugs also target this pathway. Multiple meta-analyses of human trials show berberine can reduce fasting, blood glucose, lower A1C, improve insulin sensitivity, improve lipid markers. Now here's the critical distinction. Berberine is powerful. It is not a mild herb sprinkled into tea. It interacts with medications. It can lower blood sugar significantly. It requires supervision if someone is already on glucose-lowering drugs. Mechanistically, berberine appears to improve glucose transport into cells, reduce hepatic glucose production, modulate gut microbiota, reduce inflammation. Its effects are multisystemic. What makes berberine especially interesting is that it represents a bridge between traditional medicine and modern biochemical understanding. Traditional practitioners used berberine-containing plants for centuries. Modern research isolated the compound and mapped its molecular targets. This is where plant science and pharmacology overlap. 7. Turmeric, curcumin, reducing inflammatory resistance. Insulin resistance is not just about sugar. It's about inflammation. Chronic low-grade inflammation interferes with insulin receptor signaling. When inflammatory cytokines increase, insulin becomes less effective. Turmeric contains curcumin, one of the most studied anti-inflammatory plant compounds in the world. Research suggests curcumin may improve insulin sensitivity, lower fasting, blood glucose, reduce inflammatory markers, protect pancreatic beta cells. Inflammation and oxidative stress damage insulin-producing cells over time. Curcumin's antioxidant activity may provide protective effects. There's also evidence suggesting curcumin may help prevent progression from prediabetes to type 2 diabetes in certain populations. Absorption matters with curcumin. It is poorly absorbed on its own, but bioavailability improves when taken with black pepper extract, piperine, or in specialized formulations. Turmeric is not aggressive in its glucose-lowering effect. Instead, It improves the metabolic environment, lower inflammation, better signaling, more stable regulation. Sometimes stabilizing the terrain is more powerful than forcing the outcome. 8. Holy basil, tulsi. Addressing the stress connection. Most blood sugar conversations ignore stress. That's a mistake. When you experience stress, your body releases cortisol. Cortisol increases blood sugar. It mobilizes glucose into the bloodstream to prepare for action. In short bursts, this is adaptive. Chronically, it's destabilizing. Holy basil is classified as an adaptogen. Adaptogens help regulate the body's stress response. Research suggests holy basil may reduce fasting, blood sugar, improve insulin secretion, lower cortisol levels, improve lipid profiles. Its effect may not be solely due to direct glucose interaction. It may work indirectly by calming the stress axis. This matters because chronic psychological stress is strongly associated with insulin resistance. Holy basil can be consumed as tea-dried leaf powder, standardized extract. Unlike some stronger compounds, tulsi tends to have a gentler, more systemic influence. It reminds us that blood sugar regulation is not just biochemical, it is hormonal and neurological as well. 9. Panax ginseng. Enhancing glucose uptake in muscle. When insulin is released, one of its main targets is skeletal muscle. Muscle tissue accounts for a massive portion of glucose disposal after meals. If muscle cells are insulin resistant, blood sugar remains elevated longer. Panax ginseng has been studied for its role in improving glucose metabolism. Several human trials suggest ginseng may improve post-meal glucose response, enhance insulin sensitivity, increase glucose uptake in muscle tissue, improve energy metabolism. The active compounds, called ginsenosides, appear to influence insulin signaling pathways and possibly enhance glucose transporter activity, specifically GLUT4 transporters in muscle cells. Here's why that matters. When GLUT4 transporters move to the cell surface efficiently, glucose enters muscle cells more effectively. That lowers circulating blood sugar. Unlike plants that slow digestion, ginseng works at the cellular uptake level. It doesn't block sugar, it helps use it. Some research also suggests ginseng may modestly reduce fasting glucose and improve A1C when used consistently. Different species matter. Panax ginseng, Asian ginseng, and Panax quinquefolius, American ginseng, have slightly different profiles, but both have been studied in metabolic contexts. This is not a dramatic overnight intervention. It's a metabolic modulator. For individuals whose primary issue is poor post-meal clearance, improving muscle uptake is critical, and that's where ginseng shows promise. 10. Banaba leaf, increasing cellular glucose transport. Banaba leaf comes from Lagerstroemia speciosa, traditionally used in Southeast Asia. Its key active compound, corosolic acid, has drawn attention for glucose regulation. Studies suggest banaba may increase glucose uptake into cells, improve insulin sensitivity, reduce fasting blood sugar. Some research indicates it may act similarly to insulin in certain cellular pathways, enhancing glucose transport without necessarily increasing insulin levels dramatically. Banaba appears to influence GLUT4 translocation, similar to exercise. That's an important distinction. Exercise increases GLUT4 expression in muscle cells, improving glucose uptake. If a plant compound enhances that same transporter activity, it may complement lifestyle interventions. Banaba's effect size in studies is moderate, but consistent enough to maintain scientific interest. It is often standardized in supplement form for corosolic acid content. Traditional preparation involved leaf tea, but standardized extracts provide more consistent dosing. Banaba represents another example of traditional use validated through molecular research. It works at the cellular level, not by blocking digestion, not by suppressing appetite, but by improving how cells handle glucose once it's there. 11. Nigella sativa, black seed, supporting pancreatic function. Nigella sativa, often called black seed or black cumin, has been used traditionally across the Middle East, North Africa, and South Asia. Its primary bioactive compound, thymoquinone, has been studied for anti-inflammatory, antioxidant, and metabolic effects. Research suggests black seed may reduce fasting blood glucose, improve A1C, improve insulin sensitivity, reduce oxidative stress, but one of its most interesting areas of study involves pancreatic protection. Chronic hyperglycemia and oxidative stress damage beta cells, the insulin-producing cells of the pancreas. Black seed's antioxidant properties may help protect these cells from damage. It doesn't force insulin production aggressively. Instead, it may create a healthier environment for pancreatic function. There's also evidence that black seed oil may improve lipid profiles, which often correlate with metabolic dysfunction. This is important because metabolic syndrome rarely occurs in isolation. Glucose, triglycerides, HDL, blood pressure, they often shift together. Black seed appears to influence multiple markers simultaneously. It can be consumed as oil ground seed capsules. Again, the theme emerges. Most effective plants do more than one thing. They reduce inflammation. They improve signaling. They support organ function. Systemic effects are their strength. 12. Artichoke leaf, the gut liver. Connection. Artichoke is typically associated with digestion and cholesterol, but its leaf extract has been studied for broader metabolic influence. Artichoke leaf contains compounds such as cynarin and luteolin, which appear to support bile production, improve lipid metabolism, support liver detoxification, improve insulin sensitivity. Here's why bile matters. Bile acids influence gut microbiota composition and metabolic signaling pathways. Emerging research suggests bile acid signaling is deeply connected to glucose metabolism. Artichoke's liver-supportive properties may indirectly improve glucose control. It also appears to improve post-meal lipid metabolism, which often correlates with insulin resistance. Insulin resistance doesn't just affect glucose, it affects fat handling. Artichoke leaf may improve lipid clearance, reduce oxidative stress, support hepatic insulin sensitivity. It works quietly and indirectly. Not flashy, not dramatic, but potentially meaningful over time. 14. Clove, small spice, enzyme influence. Clove is used sparingly in cooking, but it is chemically dense. It contains eugenol and other polyphenols with antioxidant and enzyme modulating properties. Emerging research suggests clove may improve insulin sensitivity, reduce oxidative stress, inhibit carbohydrate digesting enzymes. Some studies indicate clove extracts may inhibit alpha-glucosidase, the same enzyme targeted by certain pharmaceutical drugs. That means clove may help slow the conversion of carbohydrates into glucose. Because it's so potent, small amounts are sufficient. Clove doesn't function as a primary intervention. Instead, it supports a broader metabolic strategy. Its strength lies in its concentration of polyphenols. Polyphenols influence gut bacteria, inflammation, and enzyme activity. In metabolic health, small modulators add up. Clove is one of those modulators. 15. Neem, bitter, metabolically active. Neem has been used in Ayurvedic medicine for centuries. It is intensely bitter. Bitterness in plants often correlates with alkaloids and bioactive compounds. Research suggests neem leaf extract may reduce fasting blood sugar, improve glucose tolerance, enhance insulin signaling. Animal studies are stronger than human trials at this stage, but preliminary human data suggests metabolic influence. Neem may also reduce oxidative stress and inflammation. What makes neem interesting is its overlap. It influences immune function. It influences inflammation. It influences glucose metabolism. Systems overlap. Neem is usually consumed as extract due to bitterness. It's not commonly used as a food, more as a medicinal plant. The evidence is promising but less extensive than berberine or cinnamon. Still, it represents another pathway into metabolic modulation. 16. Blueberry leaf. Beyond the fruit. Most people associate blueberries with antioxidant benefits, but the leaves have their own metabolic profile. Blueberry leaf contains polyphenols that may improve insulin sensitivity, inhibit carbohydrate digesting enzymes, reduce oxidative stress. Some research suggests blueberry leaf extract may reduce post-meal glucose spikes. Like mulberry leaf, it appears to influence digestive enzymes. Additionally, polyphenols from blueberry leaf interact with gut bacteria. Healthy gut bacteria produce short-chain fatty acids which improve insulin sensitivity, reduce inflammation, support metabolic flexibility. This is where the gut connection becomes powerful. Your microbiome influences glucose metabolism, appetite regulation, fat storage, inflammation. Plants rich in polyphenols feed beneficial gut bacteria. Blueberry leaf fits that category. Consumed as tea or extract, it works gradually over time. Not forceful, but system supportive. At this point, we've seen plants that block enzymes, slow digestion, form fiber gels, feed gut bacteria, reduce inflammation, improve liver output, enhance muscle uptake, protect pancreatic cells. The gut is not separate from blood sugar. It is central. When digestion is rapid and refined carbohydrates dominate, blood sugar spikes aggressively. When fiber, enzyme modulators, and polyphenols are present, the rise is moderated, and moderation reduces insulin stress. What becomes clear across these 16 plants is this: the most effective strategies are layered. A single herb targeting one enzyme is rarely transformative, but a system built around fiber anti-inflammatory compounds, insulin sensitizers, liver support stress regulation that's comprehensive. 17. Berberine-containing plants. Barberry/Oregon, grape/golden seal. The source plants. We already discussed berberine in part 2 because it's one of the strongest researched natural compounds for metabolic regulation. But here's what many people don't realize. Berberine doesn't come from one plant. It comes from several plants, including barberry, Berberis vulgaris, Oregon grape, Mahonia aquifolium, golden seal, Hydrastis canadensis. Traditional medicine used these plants long before berberine was isolated in supplement form. Modern research suggests berberine influences blood sugar by activating AMPK, a metabolic master switch, improving insulin sensitivity, reducing glucose production in the liver, improving glucose uptake in cells, modulating gut microbiota. Now, the reason I'm listing berberine plants as its own entry is because a lot of people hear berberine and think it's just another herb. It isn't. Berberine is closer to plant-derived pharmacology. It's potent enough that it can interact with medications. So here's the responsible framing. Berberine and berberine-containing plants may be powerful supportive tools. But if someone is on insulin, metformin, sulfonylureas, or blood pressure meds, they should not just add berberine blindly. It's not a try it and see. It's a monitor and supervise. That's what makes this list credible. 18. Licorice root. Glycyrrhiza glabra, the caveat plant. Licorice root is a fascinating one because it's both promising and risky depending on the person. Certain compounds in licorice have been studied for improving insulin sensitivity, reducing inflammation, supporting metabolic regulation. But here's the catch. Licorice can increase blood pressure and alter potassium levels in some people, particularly if taken in high doses or for long periods. So why include it? because it shows a crucial truth. Natural doesn't mean harmless. Licorice root has a legitimate place in traditional medicine and has bioactive effects, but it is not something everyone should use. If someone has high blood pressure, kidney issues, heart conditions, electrolyte imbalance, licorice can be a bad idea. That's why this isn't a take everything list. It's a know what you're doing list. Licorice is here as an example of a plant with metabolic relevance and a reminder that responsibility matters. 19. Garlic, insulin support and cardiometabolic benefits. Garlic is one of the most underestimated metabolic foods on the planet. Most people think of garlic for immune support flavor, natural antibiotics. But in metabolic research, garlic is often discussed for cardiometabolic outcomes, improved lipid profiles, reduced inflammation, potential improvements in fasting blood sugar. Garlic contains sulfur compounds like allicin that influence oxidative stress and inflammation. And remember, inflammation drives insulin resistance. Garlic doesn't work like a carbohydrate blocker. It works more like a systemic stabilizer, and it has a major advantage over many supplements. It's already food, meaning it's easy to integrate consistently. Consistency is what turns small changes into measurable outcomes. If someone eats garlic regularly, especially in whole food diets, it can support broader metabolic health, not just glucose. And that matters because diabetes rarely comes alone. It often comes with high triglycerides, low HDL, high blood pressure, inflammation. Fatty liver garlic targets the bigger picture. 20. Green tea, Camellia sinensis. Metabolic efficiency through polyphenols. Green tea is more than a caffeine drink. It contains EGCG and other catechins that have been studied for improved insulin sensitivity, reduced oxidative stress, improved fat metabolism, potential improvements in fasting glucose. Green tea also influences gut microbiota composition, and it supports metabolic flexibility, the ability to shift between burning glucose and fat efficiently. Some studies show modest improvements in glucose control with consistent intake, not dramatic, but meaningful when layered with other habits. It also has one of the best effort-to-benefit ratios. It's easy, it's accessible, it's repeatable. Green tea is what you use when you want a daily baseline support habit that doesn't feel like treatment. So here's the truth. None of these plants are magic. None of them override reckless eating. None of them replace medical supervision. But they are real. They've been studied. They've been used traditionally for centuries. And many of them influence the exact biological systems that regulate blood sugar. Insulin signaling liver glucose output, muscle uptake, inflammation, stress hormones, digestive enzyme activity. Blood sugar isn't controlled by one switch, it's a network. And what you've seen in this video are tools that can support that network. Some slow absorption, some improve sensitivity, some reduce inflammation, some activate cellular energy pathways. Different mechanisms, different entry points. Which means the smart approach isn't taking everything. It's understanding your pattern. Is it post-meal spikes? Is it elevated fasting glucose? Is it chronic stress? Is it inflammation? When you know the mechanism, you choose the tool. That's strategy. And strategy beats hype every time. If you're managing blood sugar, the foundation is still. Whole food intake, fiber resistance training, sleep stress regulation. Plants don't replace those. They enhance them. And when used intelligently, especially alongside medical guidance, they can become powerful allies. This isn't about rejecting modern medicine. It's about understanding that metabolic health has always had more than one lever, and now you know 20 of them. Use that knowledge carefully, use it strategically, and use it responsibly.